Hi, it's really a pleasure to be here with all of you. Happy FD Day. It's just an honor and a pleasure to be here as part of this community, especially to hear everybody's presentations. It's such a courageous community, an inspirational community, and there's so many key figures who play such critical roles in trying to help improve the lives of people with FD. So I'm here wearing two hats. One as a neuroscientist whose lab studies FD and is interested in understanding what's going wrong at a cellular level in FD, and also developing therapeutics to potentially help members of the FD community, and also as an FD family member. So I'm all of your cousin. I grew up with two cousins who had FD. So I'm going to share my screen. So what we have been interested in is, as I said, trying to understand at a cellular level what's going on in FD. And if once we can figure out what is going wrong, can we develop ways to improve the health of neurons, which are the cells that primarily die in FD. And as Dr. Kaufman mentioned, some of these neurons are dying in utero, and some of these neurons are going to die postnatally. So, there's different strategies as to which cells we can try to rescue at different stages. But what you're looking at here in this photograph, actually in purple or in magenta, these are all the neurons in the retina that will die in FD. So this is from a mouse retina. And not all of them will die. And that's the good news, because there's many that we can save. And so the goal is, or one goal for FD is, if we can save these retinal neurons, then we could prevent the loss of vision. And that's what I'm going to be talking about today. So one way to do that is, as Dr. Kaufman mentioned, is to introduce a healthy copy of the gene that is mutated in FD. And we know that the gene has two different names. One is ELP1, ELP1, and one is IKB-KAP. And that gene is mutated in people who have FD. And so the goal is, can we improve the function of that gene? And there's different ways to do that, as Dr. Kaufman mentioned. And one way is a way we've been working with is to do what's called gene therapy, where you introduce a healthy copy of that gene. And so you put a healthy copy of that gene in a virus, and then you can inject that virus into the body, depending on what part of the body you want to rescue. And so in our case specifically, we are interested in trying to prevent the visual problems from developing. And so you can do what's called an intravitreal injection into the eye of the virus. And this method has proven already very successful for treating other retinal diseases. So for example, the FDA in the United States has already approved gene therapy for another retinal disease. And that was about, over 5 years ago, and those patients are still doing well. There's also been gene therapy has been approved for a really devastating disorder called spinal muscular atrophy, and for other disorders. And it turns out that, especially doing the eye injections, first of all, you only have to inject once over the lifetime of the person. And those injections seem to be very safe and well tolerated. And now gene therapy is in trial for numerous other genetic diseases, including other retinal disorders. So we've developed in our lab mouse models for FD, and we've now shown, and we have a paper that's been submitted for publication, that introduction of a healthy copy of the ELP1 gene in the eye of a mouse that has FD can reduce or prevent the death of about 30% of the retinal neurons. And so to us, this is proof of concept that this approach could work. And these were just using sort of off-the-shelf components, but certainly we believe this method can be optimized and develop for the clinic. So this work has been a collaboration on the gene therapy with Dr. Claudio Punzo at the University of Massachusetts Medical School. And the work was funded both by the NIH and from the generous funding of the Claire and Philip Wexler Research Fund from the Familial Dysautonomia Foundation. So thank you very much for all your support. And so I just want to show you here what this looks like in practice, because we're very excited about this. So I told you that it can save neurons, but I also want to show you what that looks like. So we did a single injection of the virus and we wait 3 months and we come back and then we look at the retina in the mouse and we count the number of retinal neurons to see, do we have more, do we have less, and what do they look like? So this picture here on the left is a picture of retinal neurons. Those in the first image I showed you, they were magenta, but here they're white. So these are the retinal neurons. And what you're seeing in red here is that they're expressing the human ELP1 protein. So this is a mouse retina, so it should not express any human ELP1 protein. But in fact, we're showing that after injecting the virus, these mouse neurons now express this human ELP1 protein. So that tells us two things. One, that we can express a healthy copy of the ELP1 gene in a retina. And two, that these are really healthy looking neurons. So that had also been a concern, that if you overexpress a protein that's not normally, that you're driving artificially, essentially, is that going to be unhealthy for the cells? But in fact, these cells that are in red here that have the arrows look very, very healthy and normal. So we are very optimistic that this project can be developed further. And so what are the next steps? So the next steps are that 2 other labs are going to get involved in this project. One is the lab of Anil Chakuri, who has been a postdoc with Sue Slagenhout. And he's going to pursue doing gene therapy in the retina. He's setting up his own lab now at Harvard. And then another collaborator, Nadia Zeltner, who has a lab at University of Georgia, and she's really interested in the autonomic crises and trying to see if gene therapy could help mitigate the severity of the autonomic crises. And so she would like to develop gene therapy to treat systemically. So I am passing the baton to these 2 people. I personally am retiring from my lab, but I'm going to stay on as co-advisor of the Scientific Advisory Board with Adrian, and very happy to try to help shepherd these projects and make sure that we can optimize this treatment as a potential therapy for FD. So lastly, I just want to say thank you to all of you for your support and to the wonderful FD community. And I hope my wonderful co-director, Adrian Gilbert, reappears soon to help steer us forward. But thanks again to all of you. You really inspire me every day.
That was so wonderful, Dr. Lefcourt. Thank you so much. And next we'll hear from Dr. Adrian Gilbert.
Thank you, Frances. I'm sorry I didn't jump in quite when I was supposed to before. I had a small Zoom crisis, but as with FD crisis, I rang the center and Abilene was very, very helpful and she sorted me out. And I'm really pleased to be back, and just shows you, no matter how many times you practice, sometimes these things just don't always work out. Anyway, thank you, Frances. That was really inspiring. I did catch the gist of your presentation. It was really, really interesting to hear. And then it's going to go forward in other laboratories. And I think now we're just going to hand over to Sue Slagenhout, who's as we all know her, who's the scientific director of the Mass General Research Institute. And she's also a professor of neurology at Harvard Medical School, who's going to talk some very exciting news on her new development of a small molecule compound for familial dysautonomia. So, Sue.
Thank you so much, Adrian. I'm glad to see you back in Zoom land with us. I loved hearing Francis's presentation and all of the presentations this morning, and I'm really pleased to be here with all of you. I have worked on FD. My lab has worked on FD for quite a long time. I think I've been talking at FD Day for perhaps 30 FD days, maybe 30 years. I came to Mass General, started working on FD in 1991, so it's been quite a long time. For those of you who have been around that long, you'll remember I used to come and talk about George. And today I'm going to share with you a little bit of the work that my lab is doing most recently. So I will share my screen. Okay, so hopefully everyone can see this. So I'm going to give you a little bit of a primer. You heard some of this from Horatio, but I want to remind you of the work that we've been doing for many, many years, and that is trying to treat something called defective splicing using a drug. So we're developing a drug to fix actually the mutation that causes FD. So you might remember that genes are broken into pieces along our DNA. And this mutation in FD, what happens is it causes these pieces not to be assembled properly. So we need piece 20 to come after 19 and before 21, like this, 19, 20, 21. But this mutation in FD causes this number 20 piece to be skipped. And what happens is we do not get any ELP1 protein made when this is skipped. We need exon 20 to be in there in order to get ELP1 protein. So my lab, ever since we first identified kinetin, has been working to find a drug that will connect in some way to this area and make sure that this exon 20 or this piece 20 is always included. And then what our goal will be is to then get more functional ELP1 protein, which you just heard a little bit about what is needed there. And so we have a drug called PTC680 that was made by a company called PTC Therapeutics, who have been great partners to both my lab and the FD Foundation for many years. They are continuing, although they're no longer developing this drug for to get it into patients. They've agreed to allow us to continue to move this forward. And so late last year, my team at Mass General and I applied for a grant from the National Institutes of Health called a Blueprint Neurotherapeutics grant. And I have number 2 on this slide because many of you who have listened to us for a number of years know that we were part of this program years ago following the identification of kinetin. We identified kinetin, and then we became part of this program to make new improved kinetin to try to make a better drug. And we've continued this work through this program and then partnered with PTC Therapeutics and identified this drug called PTC680. And this is the drug now we're trying to drive to the clinic. The title of our grant is Development of a Splice Modulator Compound for Familial Dysautonomia. So again, number 2, because we went back to the NIH and said, okay, now we have this really good drug in hand. Will you help us by forming a team and funding the work needed to hopefully get this to patients? So we have a development team that includes many people from my lab, people from the FD Center who you know, Horatio and Alejandra, NIH experts. We have drug development consultants from industry, and the team meets every other week. And we just kicked off this grant about a month ago on May 3rd. And this team meets every other week and to really do the studies and discuss what's needed to move this drug to the clinic. So what I've shown here, I don't expect you to understand what all of this means, but this table, but basically it shows you we have a lot of work to do, but we're optimistic. In year 1, we will work on various studies needed so that we can make sure that this drug, so that we can synthesize this drug in a large enough amount so that we can further test this drug in our mice and continue to work towards getting a pre-IND meeting with the FDA. And then after the first year, we move into studies IND enabling toxicology studies that will make sure that this drug is safe enough for us to give to our FD patients. We know that it works remarkably well to increase the levels of ELP1 protein, and that's our ultimate goal. And so, as I said, we're very optimistic. We have a lot of work to do, but we really hope that everything goes well. There's still, even though there's still a lot of things that could go wrong, as whenever new drugs are developed, there's many places along the way that some test or another can tell us that it's not the right drug for us. But we're very hopeful that we've already done a lot of testing on this drug, and we're hopeful that we'll be able to get it into patients. This would be an oral drug. So one of the things that we're excited about this is that this would be a drug that patients could take easily on a daily basis at home. So that's our goal with this, is if we could come up with a drug in which we could have patients be able to be dosed every day at home that would increase ELP1 protein. So I really want to call out many members of my team. This is a bigger group. This is what I call the Slog and Hep Lab team and friends, but I really want to point out Elisabetta Marini, who's here, and Anil, who you just heard about, Anil Chakuri and Daddy Gao. All three of these individuals were postdoctoral fellows, and I, like Francis, although I'm not retiring as Francis is, but we pass the torch. This is part of what we do, is train the next generation of scientists. And I feel that the FD community is remarkably lucky to have these three scientists who have now all started their own labs. They're all junior faculty members, and they've started their own labs, and they will continue the work on FD along with me. And so we're all lucky to have them, because they have varied interests, varied expertise, and will bring new ideas and new thoughts to ways that we might begin to treat FD. So we're really lucky to have all of these people who are focused on FD. So again, I look forward to answering any questions that you might have, and I'm thrilled to be part of FD Day for so many years. And with that, I will end and turn it back to Adrian.
Thank you very much, Sue.